students. Today we will learn about environmental biotechnology. So, before going to study about environmental biotechnology, here we will see a glance on environment. Environment includes earth, water, atmosphere and space. So, today we are polluting a lot of environment with pollutants which is harmful to our daily life. Environmental pollution includes three main types, mainly atmospheric pollution, soil pollution, water pollution. So, today we will discuss about the water pollution. Water pollution is mainly caused due to domestic effluents and industrial effluents. So, if you see this industrial pollution, the effluents are contaminating the surface water bodies and they are releasing water gases, thus contaminating both air, land and water. Dumping of unwanted things result in adding million tons of hazardous waste into soil and groundwater. Liquid and solid waste are added to environmental by accidental spillage and leakage during transport of chemicals. These are all causing pollution to our environment. So, remediation, what we have to do? Remediation of contaminated land and groundwater is needed, and so the biotechnology here plays or contributes in treatment of such sites by using indigenous microflora or added specific microorganisms which help in biodegradation of pollutants and recovery of land and groundwater. If you see the causes of contamination, the pollutants that may occur due to draining of liquid waste on open land, dumping waste, sludge on unused plots, accidental leakage of storage tanks or underground pipelines, or spoilage from tankers on land or in water bodies like rivers, seas and oceans. So, what are these hazardous chemicals released due to the pollution? Refractory organics, oils, benzene, styrene, vinyl chloride, pentachlorophenols and polyaromatic hydrocarbons, toluene, xylene and phenols. These are all the hazardous chemicals which are going to be released in water. So, how the biotechnology plays a role in protecting the environment? If you see the definition of biotechnology, it is nothing but integration of natural sciences and engineering in order to achieve the application of organisms, cell parts thereof and molecular analogues for products and services. This definition was given by EFB in 1989. So, what is this environmental biotechnology? It is nothing but the application of the process for protection and restoration of quality of our environment, and it is the solving of environmental problems through the application of biotechnology. And why we have to study environmental biotechnology? It is needed to eliminate the hazardous waste produced by other technologies, and here we can distinguish between similar species and ensure those species not at risk of extinction, and it is needed to create alternative energy sources like biofuel. So, if you see biotechnology in the presence of living organisms, here we are able to recover the contaminated land and contaminated groundwater. What are the strategies for clean environments? So, here there are two main strategies. One is bioremediation and second one is biodegradation. So, if you see biodegradation, here compounds are there, microorganism is responsible, and to grow that particular microorganism, environmental conditions should be safeguarded. So, these are all the compound to be degraded is necessary, and the microorganism which is degrading the compound here is nothing but the hazardous chemical, and the environmental conditions should also be taken into consideration so that that particular microorganism can grow and which can degrade the compound by its metabolic activity. So, if you see the definition of bioremediation, it is the stimulation of living organisms to rapidly degrade hazardous organic contaminants to environmentally safe levels in soils, subsurface materials, water, sludge, residues etc. So, these main points in this bioremediation are use of biological systems. Bio means nothing but living system. So, the biological system for the reduction of pollution from air, aquatic or terrestrial systems. This process involves aerobic and anaerobic, which we will discuss later in detail. Microorganisms that result in degraded products, and it is also used to reduce or remove hazardous waste which has already polluted the environment. So, this is the mechanism through which, here if you see, the microorganism is degrading the pollutant and the degraded product is getting converted as eco-friendly substance. So, what are the requirements for this biodegradation or bioremediation? So, we have to provide conditions in contaminated environment so that appropriate organism flourish and carry out the metabolic activities to detoxify the contaminants. Using contaminants as nutrients or energy source, the microorganisms are degrading the contaminants by their co-metabolism. So, where can we apply this bioremediation? In wastewater and industrial effluents, drinking and processing water, soil and land treatment and solid waste. If you see this wastewater and industrial effluents, we can remove common pollutants from water before discharge into river or sea. The elimination of phosphorus, nitrogen, heavy metals and chlorinated compounds can be completely eliminated. Sulfur reducing bacteria can be used to remove waste containing sulfur, and the bed containing waste material and microbes were placed in suspension bioreactors. Thereby, we can degrade the pollutant. If you see the drinking and processed water, biotechnology for reclamation and purification of wastewater for reuse, agriculture regions of the world, animal waste and excess fertilizers are resulting in high level nitrates in drinking water. So, this can be clarified, and biotechnology has provided successful methods by which these compounds can be removed from processed water before it is delivered to customers. If you see the soil and land treatment, here there are two types, in situ and ex situ, which is in situation and outside the situation. Bioaugmentation is nothing but the introduction of microorganism in that situation or in that place, and biostimulation is nothing but ventilation or adding nutrient solution to promote the bacterial growth. So, ex situ means we are bringing the soil, contaminated soil or groundwater and treating it in separate laboratory conditions for testing the effect of the organism on that pollutant. If you see the solid waste, these solid wastes are composed of readily biodegradable organics. And what are these waste? The waste can be converted to valuable source. So, unless and until it is converted into valuable resource, need of bioremediation is nothing by composting or anaerobic digestion. What is this anaerobic digestion? Which permit the recovery of substantial amounts of high value biogas together with high quality stable organic residue in the absence of oxygen. Anaerobic means in the absence of oxygen. The organic residue now can be used as manure, and thus recycling of solid waste can save enormous cost of incineration. So, here I have shown, you see river Ganga is polluted with domestic sewage and industrial effluents. These are the pipelines through which the industrial effluents and domestic sewage is released into the river water. So, this is the process of bioremediation, wherein the contamination is sent here and we are supplying oxygen and nutrients, means it is aerobic, and here carbon dioxide is released. We are injecting the contaminants through this pipeline and we are recovering the biodegraded and useful material here, we are recovering here. So, what are the organisms, microorganisms which are used for bioremediation? Generally, we see that bacteria, fungi and plants help in bioremediation. If plants are used, we are calling it as phytoremediation. If fungi is used, we are calling it as mycoremediation, because study of fungi is nothing but mycology. And next one is bacterial or microbial bioremediation. All these are living organisms, so these are categorized under bioremediation. So, Pseudomonas, Mycobacterium, Alkaligens, Nocardia, Arthrobacter, Cornybacterium species, Bacillus species, and Candida, Aspergillus, this is fungus, and Xanthomonas, this is bacteria. So, these are the main organisms which are used in biodegradation or bioremediation. So, this is a type of bioremediation system. We are here using Bioslurry, where sulfur, sludge of the soil is taken, and here we are subjecting it into, passing it into Bioslurry reactor, and from there here it is clarified. So, we are calling it as a clarifier, and it is discharged outside, and thus the Bioslurry bioremediation system works. For wastewater treatment, here this is the process where the first stage converts the organisms into methane, the products organics into methane, that is the pollutants are converted into methane, and second stage is the one where the nutrients are utilized. What are the factors that affect biodegradation? So, chemical nature of xenobiotic means what? Nothing but hazardous chemicals, we are calling it as xenobiotics, that has been degraded, and capability of individual microorganism, nutrients and oxygen supply, temperature, pH, redox potential, biostimulation and bioaugmentation. So, these are the main factors that affect the biodegradation mechanism. One is the chemical nature of that component or pollutant, and second one is the individual organism, nutrient and oxygen supply. In order to grow that particular organism, they require nutrient and oxygen supply. So, it also depends on this nutrient and oxygen supply of that particular microorganism, and the temperature levels, pH levels and redox potential levels are also taken into consideration. And if you see biostimulation and bioaugmentation, these are the two approaches or developments to enhance biodegradation by microorganisms. Then what is the difference between these two, biostimulation and bioaugmentation? Biostimulation is the process in which the microbial activity is enhanced by increased supply of nutrients. Already the microorganism is surviving there, and we are enhancing its growth or promoting its growth by increased supply of nutrients or by addition of certain stimulating agents like electron acceptors and surfactants. So, this is about the stimulation, that is we are stimulating the growth of that microbia or microorganism by increasing supply of nutrients or by adding stimulating agents. That is why we are calling it as biostimulation. And bioaugmentation, which is used to increase biodegradation through manipulation of genes by employing a consortium. Consortium is nothing but a group of microorganisms, not only the single individual, but if they combine in a group of organisms, they are showing the more amount of degradation. So, that we call it as augmentation. Bioaugmentation is nothing but manipulation of genes by employing a consortium of microorganisms. So, here we will see the definition of bioaugmentation. The bioaugmentation is nothing but process of seeding contaminated sites with the competent microflora produced in fermenters. That is, we are growing the microflora in fermenters, and that those microorganism growth or microbial growth we are using to speed up that mechanism of bioremediation. So, the seeding contaminated sites means nothing but polluted sites with a competent microflora produced in fermenters and used to speed up bioremediation is called bioaugmentation. So, it is done in these following cases. For recently contaminated sites by spills, oil spills, contamination with a poorly biodegradable compound. So, that compound is poorly biodegradable, at that time we can use this bioremediation, and when toxic metal concentration is high. So, and where rapid cleanup is required, if indigenous microflora degrades contaminant only partially to toxic intermediary level, then we can apply this consortium of microflora. When adaptation to xenobiotic recalcitrants is slow, recalcitrant. So, xenobiotic is nothing but chemical, hazardous chemical which is not degraded easily, that which is not recovered easily, degraded easily, it is called as recalcitrant. So, here we use three main parts, three main steps. Here one is bioreactors and second one is enzyme responsible for that microorganism, that is the metabolic activity. To promote the metabolic activity in the microorganism, what are the enzymes required? So, first we will see about the bioreactors for bioremediation. Here two main approaches are there, one is in situ operation and second one is ex situ operation. So, polluted zone compartment is subjected to restoration in situ operation, whereas in ex situ operation the contaminated isolated volume of material that is undergoing the treatment on site. So, this is in situation where the contamination is there we are going and applying, and ex situ means we are bringing the contaminants into the laboratory conditions and we are applying against microbial growth. So, here three types of bioreactors are used in bioremediation, namely static beds and heaps of soils where intermittently they are irrigated, aerated and mixed, and rotating cylindrical and horizontal screw mixed type of bioreactors where continuous aeration, mixing and irrigation takes place. And third one is a different one where we are using aqueous slurry subjected to aeration and agitation. Agitation is nothing but mixing. And what are the factors which assess the right process in bioreactors? Number 1, process economics, safety, system reliability, operational flexibility, acceptable residue concentration of pollutants after treatment, and these are the main factors in assessing the right process in bioreactor. So, for what we are using agitation? Agitation enhances the process rate and avoids separation. Segregation is nothing but separation, and heterogeneity of microbes exists both in inside and offsite. So, we have seen bioreactors till now. Now, our second part is enzyme systems for biodegradation. So, generally we know that genes encoding enzymes of biodegradation are present in both chromosomal DNA and plasmid DNA. Here we have listed some xenobiotic compounds and the name of the plasmid on which the enzyme system is working. This is only on Pseudomonas. Pseudomonas is the major microorganism which helps in biodegradation or bioremediation. See here, if you see the xenobiotic is naphthalene. Naphthalene, for that compound the plasmid, that is genes encoding enzymes of biodegradation are present on the plasmid NaH. We have taken the code as NaH. Xylene XYL. Xylene and toluene, TOL for toluene and XYL for xylene, and salicylate SAL, camphor CAM, and 3-chlorobenzene. These are the main pollutants or hazardous chemicals or xenobiotics which are present in oil which is leaked in the water. So, here we can see the list of microorganisms and the xenobiotics degraded. First one, if you take Pseudomonas, it can degrade the aliphatic and aromatic hydrocarbons like naphthalene, benzene, toluene, anthracene, xylene, malathion and parathion, or the bioinsecticides and biopesticides. Next, Mycobacterium species, which is degrading benzene, cycloparaffins and branched hydrocarbons. Alkaligens species, it degrades PCBs and halogenated hydrocarbons. And Nocardia species, naphthalene, alkyl benzene and phenoxyacetate. Arthrobacter species, benzene, polycyclic aromatics and phenoxyacetate. If you see here another one, Corynebacterium species, it is degrading the halogenated hydrocarbon to which the halogen is attached is the halogenated hydrocarbon and phenoxyacetate compounds. The Bacillus species can degrade the long chain alkenes and phenylurea. Candida species, PCBs, and Aspergillus species, phenols, and Xanthomonas species, polycyclic hydrocarbons. So, these are the 10 microorganisms out of 40 to 50 microorganisms which can degrade more number of xenobiotics. Coming to types of bioremediation. So, we can classify the bioremediation types based on the site and based on the type of reaction. So, based on the site, we are categorizing it into in situ and ex situ. In situation, in site means in situ, ex situ means off site. Based on types of reactions, whether utilizing oxygen or not, here we are categorizing into aerobic, anaerobic and sequential. So, if you see the in situ bioremediation, which is categorized based on the site, it is a direct approach for microbial degradation of xenobiotics at the sites of pollution and addition of adequate quantities of nutrients at the sites. We are taking the nutrient to the particular site which to promote the microbial growth. In the presence of pollutants, they develop metabolic activity to degrade them. So, that mechanism is nothing but biodegradation. For example, this can be applied where we need to clean up oil spillages and beaches. And what are the advantages of this? It is cost effective, minimal exposure to public or site personnel. So, we are not bringing the contamination to our lab. There, wherever contaminated site is there, we are going and applying. So, we can reduce the exposure of that contaminant to public and site personnel, and sites of bioremediation remain minimally disrupted. If you see the disadvantages, it is very time consuming process and the sites are directly exposed to environmental factors where the microbial growth differs from season to season. So, based on the temperature, oxygen supply, these are the environmental factors which are affecting the microbial growth. So, the sites are directly exposed. This is the disadvantage of in situ bioremediation. Microbial degrading ability varies economically, and if you see the ex situ bioremediation, we are collecting the waste or toxic materials from polluted sites, and bioremediation is done with requisite organism carried out at designed place, and it is an improvement over in situ type. So, an improvement over in situ type, what we are doing here, we are collecting the waste or toxic materials from polluted sites. Next one, we are using the requisite organism which is degrading that particular chemical at the designed place, and it is the more improved one. If you see the advantages, number one, it is better controlled and more efficient process, and process can be improved by enrichment with desired organism, desired microbe, and it is relatively short time. In relatively short time, we can identify the particular organism which is going to degrade that particular xenobiotic. And what are the disadvantages? If you see disadvantages of ex situ, it is very costly process and the sites of pollution are highly disturbed. Why? Because we are bringing that contaminant into our lab. So, it is highly disturbed and there may be disposal problem. So, after completion of the process, where to dispose them? If we dispose them, again it may contaminate that particular site. So, these two are the ex situ and in situ bioremediation types based on the sites. Now, based on the utilization or reaction steps, we are categorizing the bioremediation types into 3 types: aerobic, anaerobic and sequential. If you see this aerobic, utilization of oxygen for oxidation of organic compounds, and what are the enzymes involved here? Monooxygenases and dioxygenases. So, they act on both aliphatic and aromatic compounds, and dioxygenases help on aliphatic compounds only. If you see anaerobic, it does not require any oxygen and growth of microbes and degradation processes are slow here and it is cost effective. If you see sequential, here under sequential we can study the biodegradation of hydrocarbons and biodegradation of other toxic compounds. Under hydrocarbons, we are categorizing them into aliphatic hydrocarbons and aromatic hydrocarbons, and under aromatic hydrocarbons also halogenated aromatic compounds and non-halogenated aromatic compounds, we are categorizing them. And these toxic compounds are nothing but the pesticides and insecticides. So, biodegradation, if you see the first one, biodegradation of hydrocarbons, they are the main pollutants from oil refineries and oil spills, and it can be possible by a consortium or cocktail of microorganisms. That is, consortium means nothing but a group of microorganisms. Best example is Pseudomonas, Corynebacterium, Orthobacter, Mycobacterium and Nocardia. So, here if you see this diagram shows hydrocarbon oxidizing bacteria that attach to oil slicks. So, whenever oil is Gulf oil, we have seen one situation where the oil is released into the ocean. So, at that situation we are using the carbon oxidizing bacteria and it is eating the petroleum or oil slicks. Here we can clearly see microbes eating Gulf oil and microbes present in the oil slick. So, what are the reactions here we are using? One is hydrogenation and dehydrogenation and dehalogenation, and third one is carboxylation and decarboxylation, where we have to apply hydrogenation and dehydrogenation is applicable to degrade benzoate, phenol, catechol, and dehalogenation for PCBs and chlorinated ethylenes. Chlorination, what is chlorination? Addition of chlorine to the particular compound is called chlorination, and carboxylation and decarboxylation can be used, reactions can be used in degrading toluene, cresol and benzoate. So, biodegradation of aliphatic hydrocarbons. If you see the aliphatic hydrocarbons, uptake of these is a slow process due to their low solubility in aqueous medium. Unsaturated hydrocarbons are degraded in both aerobic and anaerobic environments, and saturated hydrocarbons are degraded by aerobic process. So, saturated hydrocarbons are degraded by only aerobic process and unsaturated hydrocarbons can be degraded in both situations. Aliphatic compounds recalcitrant to aerobic processes are effectively degraded in anaerobic environment.